Hello, everyone, and welcome to today's webinar titled Selective Depletion of CD45RA Expressing Cells to Enrich Antigen-Specific T Cells for Immunotherapy. This webinar is a part of the 10th Annual Cancer Research and Immuno-Oncology Week. I am Antonina Salcido of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots and sponsored by Miltenyi Biotech. For more information about Miltenyi Biotech, please go to miltenyibiotech.com. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Sandhya Sharma, Research Technologist in the Laboratory of Clenora M. Rooney. For a complete biography on our speaker, please visit the Biography tab at the left of your screen. Dr. Sharma, you may now begin your presentation.
Hi everyone, thank you for tuning in. And today I'll be presenting our work in enhancing and enriching antigen-specific T cells for immunotherapy, and whereby we have developed an approach to do so. These are my disclosures, and I would also like to say special thanks to Miltenyi Biotech for their invitation and this opportunity to present our work here today. So we all know about adaptive cellular immunotherapy. It's really using immune cells to fight off disease. And today we will focus on virus-specific T cells, or VSTs, which do so by recognizing viral epitopes presented on target cells. And in doing so, we utilize virus-specific T cells in post-transplant infection or against viral malignancies. For post-transplant infection, it is used against viral reactivation and infection, where I've listed all these viruses, as you can see here: Epstein-Barr virus, CMV, adenovirus, BK virus, or many more other viruses. This is mostly used in allogenic donor-derived VST setting. And next, against viral malignancies, we know that 10 to 12% cancers worldwide are oncogenic viruses. And I have listed here the 5 most oncogenic viruses, as you can see here. And over the past few years, there have been new developments and effective treatment against these viral-associated malignancies in both autologous and allogenic settings. So today we'll really focus on viral malignancies and virus-specific T cells used against those. So the VSTs that we use against viral malignancies can either be engineered VSTs, which really means that they have transgenic T cell receptor, whereby the transgenic T cell targets a single epitope on single target antigen. However, the downside to this is that if the tumor downregulates HLA or there is an antigen loss in the target cell, then the effectiveness of this transgenic TCR expressing virus-specific T cells is going to be diminished. So what we will focus on today is virus-specific T cells with native T cell receptors, whereby the VSTs can be expanded to multiple epitopes targeting multiple antigens in target cells. And this started with this initial study where EBV-specific T cells were used to target EBV-positive post-transplant proliferative disease, PTLD, and were quite effective. Ever since then, there have been subsequent studies, as I have listed here, against EBV, HPV, MCP gamma virus, where these have validated the effectiveness of virus-specific T cells or their potential with native TCR in autologous settings. So in using these virus-specific T cells against virus-associated malignancies, you can either use them as tumor-infiltrating lymphocytes, which we know have been used in HPV. However, the problem with using TILs is that it is really difficult in manufacturing and accessing these tumor-infiltrating lymphocytes. The next way you can utilize these virus-specific T cells is by isolating and generating them from peripheral blood, which we know have been used in EBV-MSV gamma virus. So, however, but then again, there is this problem that manufacturing from peripheral blood are still limited by difficulties in expanding these virus-specific T cells from patients in autologous settings, especially because these T cells that are circulating in the peripheral blood might really be rendered dysfunctional in suppressive tumor microenvironment, or it could just be that they are low in frequency in circulation. So today in this webinar, we're gonna, in terms of three main points, we're gonna talk about our objectives. The first one being understanding the obstacles to successful manufacturing of antigen-specific T cells in immunotherapy. The next one will be highlighting the effects of CD45RA depletion, which is our approach, and I will expand on it in a bit. And finally, understanding the effects of our approach and its implications in immunotherapy. So let's begin with understanding the obstacles, as I mentioned before, that when we are generating cells from peripheral blood in patients, there could be problems where these cells are just low in frequency and are dysfunctional. So in order to study this obstacle, we utilized our model of Epstein-Barr virus specific T cells, or EBVSTs as we call them, in our clinical trial against EBV-positive lymphoma. We know that 30% of Hodgkin's, non-Hodgkin's lymphoma are Epstein-Barr virus positive. And as you can see here, that the EBV-positive lymphoma B cell expresses the latent viral antigens as shown here. And we can generate T cells that would target this EBV-positive lymphoma B cell by targeting these EBV viral antigens as shown here. And these cells are called EBV-specific T cells. And these are the T cells that are expressing native T cell receptors specific to these EBV antigens. And how we do so is basically just in a graphical representation. You draw the blood from the patient, then you harvest or isolate these peripheral blood mononuclear cells, as we call them PBMCs. These PBMCs are then stimulated with EBV pep mix, which are really overlapping peptide libraries of the 4 EBV target antigens that I mentioned earlier, which then results in selective expansion of the T cells that are responding to that EBV antigen stimulation. In more details, so you have your PBMCs, then you provide these EBV peptide libraries or EBV pep mixes. The cells which recognize those EBV antigen and are then going to be stimulated and expand. These cells are then further stimulated with the antigen-presenting cells or other co-stimulated cells to specifically expand these cells that responded. Then you get your EBV-specific T cells. These EBV-specific T cells are then further adaptively transferred into the patient so that they can target and kill EBV-positive lymphoma cells. And so when we looked into our clinical trial, the EBV-specific T cells, we noticed that we had 27.5% manufacturing failure. And what that really means is that we noticed that 27% of the lines that were procured from the patients either failed to expand, or even if they expanded, they just lacked EBV antigen specificity. So we further looked into this phenomenon of manufacturing failures, and we also noticed this manufacturing failures in HPV-specific T cells, which has also been generated in our clinical trial under Dr. Carlos Ramos and his clinical trial. So for our EBV-specific T cells, since we saw 27% manufacturing failures, we went and looked into the lines that we have generated, the EBV-specific T cell lines that were generated from the patient. What you're seeing here is a waterfall plot where we're looking into the interferon gamma released by the T cells in response to EBV antigen stimulation by ELISpot assay. On the y-axis is the number of T cells or interferon gamma spot forming cells, which for every 100,000 EBV-specific T cells that secreted interferon gamma when they're stimulated with EBV antigen. On the x-axis, each line represents a patient line that was attempted to generate. And you can notice here that this dotted line, which signifies as less than 50 SFCs, are the lines that only secreted 50 cells that had secreted interferon gamma over 100,000 EBV-specific T cells. And you can see here that a lot of the lines had really low antigen specificity, below 50 spot-forming cells. This just tells us that a lot of our lines do lack antigen specificity. When we specifically looked into these lines, you can notice here that a lot of the lines, they just do not grow. We know that. And then some of the lines, they do grow, but then they still lack EBV specificity. So because we see that they grow and they lack EBV specificity, we looked into our lines phenotype. And as you can see here, that a lot of our lines, even though they did have CD3 positive T cells, you can see that a lot of them have a very high frequency of CD56 positive, CD3 negative NK cells. So this tells us that in addition to expanding CD3 positive T cells, we are also expanding other cells like NK cells in our culture, which could be one of the reasons why our cells lack EBV antigen specificity. So from our observation, we're able to identify two major problems: that some lines do not proliferate, but when some lines do proliferate, they do still lack EBV antigen specificity, and that there is presence of unwanted or non-EBV specific bystander cells, which led us to then ask, can we specifically expand antigen-specific T cells from peripheral blood? So we know that the PBMC is composed of myeloid cells and lymphoid cells. And what we are really interested is in expanding these T cells residing in lymphoid compartment to generate our EBV-specific T cells. And further looking into the T cells, we know that T cells can be characterized and divided by various surface marker expression. And one of the most commonly associated marker is CD45. And we know that the naive cells and the stem cell-like memory cells reside in CD45RA positive fractions. The memory cells reside in CD45RA positive fraction. However, the terminally differentiated memory cells, which are supposed to be high effector but short-lived, reside in RA positive fraction again. So we know that the RA positive has the memory cells, and we know that after viral infection resolution, the T cells reside in memory compartment. And then this CD45RA positive fraction really includes this naive TSCM and T effector memory, which is not of so much interest to us in generating memory residing antigen-specific T cells. However, CD45RA positive PBMCs also comprises regulatory T cells, B cells, and NK cells, which are also not the cells that we want in our culture when we're generating T cells for our patients. So with this, we rationalized that if we were to deplete this CD45RA positive expressing PBMC subset, it will decrease our NK cell population and bystander cell frequency and enrich memory residing T cells, which will then result in enrichment of our memory residing EBV specific T cells. So with that, we hypothesized that selective enrichment of memory T cells will enhance the antigen specificity of our EBV specific T cells. So for that, what we did was we took our PBMCs, as I mentioned before, then we selectively removed the RA positive cells from the PBMCs and then resulted with these memory enriched or RA depleted PBMCs. These RA depleted PBMCs were then stimulated with EBV PEP mixes to generate our memory enriched RA depleted EBVSTs, which for now we'll be calling them RA depleted EBVSTs or RAD EBVSTs. And we did this CD45RA depletion by utilizing human CD45RA microbeads available from Miltenyi Biotech. And how this magnetic leveling works is that these humans CD45 antibody is attached to these magnetic microbeads. Any cells that is expressing CD45RA, when incubated with these microbeads, is going to get attached to these microbeads. So when you run these cells through this column attached to this magnetic platform, every single cell that is attached to this microbead is then going to get stuck in the column. However, then we get this elute, which is depleted of RA positive expressing cells, which are stuck in the column. So with this technique, then you get this eluted CD45RA depleted cells, which is highly enriched in memory cells. And we did test the system with both CD45RA depletion or just by selecting CD45RA positive cells and using these cells for our culture. So now that we know the system and we started doing this, first we wanted to know how effective is utilizing this CD45RA microbeads in order to remove CD45RA expressing cells. As you can notice here that for this donor, we had about 62% of CD45RA positive cells in whole PBMCs. And by utilizing this multi-biotic microbeads, you can notice that it was highly effectively depleted to just leave 0.03% of CD45RA positive cells in our RA depleted PBMC fraction. This is more clearly shown in this graph for all the other donors combined, and you can see that we are indeed able to achieve highly successful and clean depletion of RA positive cells with utilizing this technique. In addition, as I mentioned before, that presence of NK cells was also one of the problems, and that NK cells do reside in RA positive fraction, as you can see from this dot plot in the lower part of the slide, where you see that there was about 7.3% of NK cells did indeed were CD45RA positive NK cells, and the depletion of RA positive does also deplete NK cells in our resulting RA depleted PBMCs, as you can notice here. So now that we know that we are able to effectively deplete CD45RA positive cells and understand that lack of antigen specificity and or diminished proliferation is one of the obstacles to successful manufacturing of antigen specific T cells in immunotherapy, I want to further touch another point of highlighting the effects of this RA depletion. So now we did RA depletion. Our ultimate goal is looking at characterizing the functionality of our T cells. So first, we wanted to look into the proliferation of our T cells, where we compare the proliferation of whole EBVSTs, shown here in black, with the RA-depleted EBVSTs, shown here in red. And we were able to demonstrate that RA-depleted EBVSTs do indeed demonstrate higher fold expansion compared to what we observe with whole EBV-specific T cells over 16 days of EBV antigen stimulation. And these were all done in healthy EBV-positive, seropositive, EBV-seropositive healthy donors. So now that we know that the cells are able to proliferate, our ultimate goal was looking into the antigen specificity. So we looked into the antigen specificity in our RA-depleted EBVSTs, and as shown here by D for RA-depleted EBVSTs and W for whole EBV-specific T cells, you can observe that RA depletion really does significantly enhance the frequency of antigen-specific T cells as denoted by interferon gamma releasing T cells by ELISpot assay compared to what you observe with whole EBV-specific T cells in these three representative donors. When we look into the overall donors, you can also see that in the donors that we have looked so far, we do significantly enhance the overall antigen specificity of our RA-depleted EBVSTs compared to whole EBV-specific T cells. Now that we know that it enhances the interferon gamma, we then wanted to look at the polyfunctional T cells by looking at interferon gamma and TNF-alpha by our intracellular cytokine staining assay. As you can see here, that on the top panel, you are looking at the interferon gamma and TNF-alpha, some secreted by the whole EBV-specific T cells, and in the bottom by the RA-depleted EBVSTs. And you can observe that RA-depleted EBVSTs with EBV stimulation do indeed secrete significantly high frequency of interferon gamma and TNF alpha T cells compared to what you observe with whole EBV-specific T cells. And this is also further shown in this graph, where you do see that statistical difference in the increased frequency of polyfunctional cells as defined by interferon gamma and TNF alpha positive cells in our assay. So now that we know that it secretes more interferon gamma, it secretes more TNF alpha, we really wanted to look at the target antigen-specific cytotoxicity. And for that, we did a chromium release assay. And as you can see from our percent-specific lysis graph, that RA-depleted EBVSTs, shown here in red, do demonstrate enhanced percentage of killing against EBV-positive targets, shown in the left, compared to what we observe with whole EBV-specific T cells. And I would also point out to the right of that graph, you can notice that when these T cells are incubated along with the non-EBV positive targets, they do not kill, showing that there is no autologous killing of the target cells. So next, we really wanted to look at the phenotype and the frequency of the T cells that are comprising our EBV-specific T cells. As you can see here, for RA-depleted EBVSTs in red, that they do indeed have a higher frequency of CD3-positive T cells and decreased frequency of CD3-negative, CD56-positive NK cells in our culture. And interestingly, we were also able to identify that with our RA-depleted EBVSTs, we observed this more balanced ratio of CD4-positive T cells and CD8-positive T cells compared to highly skewed CD8-positive T cell in our whole EBV-specific T cells. Though we did not delve much further into what this means and how this affects our T cells, there have been various publications and there have been various research in the past year, over various years, to indeed show that the presence of CD4-positive T cells does help in the efficacy and potency of our T cell products in immunotherapy. And we do believe that this balanced ratio of CD4 and CD8 does indeed contribute to the increased antigen specificity and potency of our T cells. So now I've so far shown you that with CD45RA depletion, we are able to demonstrate, compared to whole EBVSTs, that they demonstrate enhanced proliferation, antigen specificity, target antigen specific cytotoxicity, and increased frequency of polyfunctional cells. So now that we know that they do have high frequency, I would like to go back to the same graph that I showed earlier about T cells and point out to this fact that there is also this stem cell-like memory in the CD45RA positive fraction, which is really these cells which have been identified and shown to have long-term persistence, proliferation, and stem cell-like functionality. So there is a concern that is the removal of these RA positive cells diminishing our long-term potential and functionality. So we asked, how about long-term functionality and persistence of our RA-depleted EBV-specific T cells? And in order to do so, first we looked into the phenotypic subsets of our T cells. And when we compared the T central memory subsets in our whole and RA-depleted EBVSTs, as you can see here, that there was no statistically significant differences in the central memory population in our whole versus RA-depleted EBV-specific T cells. Additionally, what we did observe was that our RAD-EBV-specific T cells do actually demonstrate lower frequency of TIM3, KLRG, and LAG3 positive T cells compared to whole EBVSTs, and these are the T cell markers that have been associated with T cell exhaustion in immunotherapy. And when we just look into the overall frequency, it is very clear in this graph that RA-depleted EBVSTs do indeed have lower frequency of this exhaustion marker expressing T cells compared to whole EBV-specific T cells. Then, after we know that there is some phenotypic differences, we did want to look into the long-term proliferation. So, in order to look into that long-term in vitro proliferation, we provided simultaneous antigen stimulation every day 7 after day 9 and looked into the proliferation of our T cells. As you can notice here, that with the with each antigen stimulation, RAD EBVSTs in red do continue to proliferate with much higher expansion fold than what you observe with whole EBV-specific T cells. And now that we know that they do proliferate, looking into their antigen specificity, you can notice here that RA-depleted EBVSTs do indeed demonstrate higher frequency of antigen-specific T cells than what you observe with whole EBV-specific T cells. And even though it decreases by day 30, you can notice that it is still higher than what you would observe with whole EBV-specific T cells. So now that we know that they are persisting for longer period in vitro, we wanted to do the ultimate test of testing in vivo. So for that, we established a EBV transformed B cell line, which are B cell line transformed with EBV and subcutaneous tumor in our NSC mice murine model. After the tumor was established, we then adaptively transferred our whole PBMCs, EBVSTs, or EBVSTs generated from RA-depleted PBMCs, and then measured the tumor. As you can see here, so the tumors were also labeled with GFP firefly luciferase, so we can also identify their bioluminescence. And as you can see from this bioluminescence imaging, that around day 12, when the whole EBV-specific T cells still demonstrate quite a bit of tumor, the mice receiving RA-depleted EBVSTs have heavily diminished bioluminescence. This is also corroborates with our measuring of the tumor volume, as you can see here from this graph for whole EBV-specific T cells shown in black compared to control group in green. And the mice that received RA-depleted EBVSTs shown here in red do demonstrate that rapid control of the tumor that you cannot observe with whole EBV-specific T cells. Even though both group whole EBVSTs in black and RA-EBVSTs in red do go on to clear tumor at the end, you should be able to notice this early rapid control of tumor by our RA-depleted EBVSTs, which also is related with this decreased metastatic tumor burden in this, as shown in this bioluminescence imaging. You can notice that our mice that received RA-depleted EBVSTs lack that metastatic tumor, which is observed in whole EBVSTs. And this rapid tumor control will be important in patients where we are really trying to control high physiological tumor burden. So now that we have shown this in our in vitro model and in our in vivo model with our EBV seropositive healthy donors, we really wanted to go to our ultimate goal of translation. What about in patient PBMCs? So in order to look into that, we generated whole and RA-depleted EBVSTs from patient PBMCs who were enrolled in our clinical trial. And as you can notice here that RA-depleted EBVSTs do indeed demonstrate high antigen specificity compared to what you observe with whole EBV-specific T cells. And over very different patients that we looked into, we were able to see the same phenomenon. And I would like to specifically point out patient 2, patient 5, and patient 6, where you can appreciate that in where they lacked any antigen specificity and we were not able to generate these EBV-specific T cell lines. Now we are able to successfully generate EBV-specific T cell lines with our RA depletion approach. Thus, we know that we can indeed restore antigen responsiveness and successfully generate EBVSTs in patients that previously failed to grow. So, in addition to just increasing antigen specificity, as I mentioned before, high frequency of NK cells was another problem in our culture. So when we looked into the NK cells, you can see over here that the RA-depleted EBVSTs do indeed decrease the resulting NK cell frequency in our EBV-specific T cells compared to what you observe in whole EBV-specific T cells. So with these observations, we were able to find this clinically feasible approach in generating potent EBV-specific T cells. And since the Miltenyi CD45RA microbeads are clinical grade, they are GMP compliant and can be used for clinical translation. There is only barely like 20% cell loss during the actual process, and that can really be overcome by the enhanced proliferation of RA-depleted EBVSTs. So it is highly feasible even in patients where sometimes you might have low number of total PBMCs count in these patients. And in addition to showing that we were able to generate highly enriched antigen-specific T cells for EBV-specific T cells, CD45RA depletion is also applicable to generating virus-specific T cells specific to other viruses like VZV, HPV, adeno, BKB. And here today, I will just show you an example of us generating virus-specific T cells specific to adeno and VZV. And you can see here that compared to whole, RA-depleted VSTs for both adeno and VZV demonstrate higher frequency of antigen-specific T cells, just still demonstrating that this platform can be applicable to generating virus-specific T cells. So this is a very feasible platform for generating clinically potent VSTs. And as so, we have successfully translated this approach into 2 of our clinical trial against using EBV-specific T cells, as we have amended our EBVSTs clinical trial against refractory relapse EBV-positive lymphoma in autologous setting, and also in a second clinical trial where we're using chimeric antigen receptor with modified EBV-specific T cells against lymphoma in allo setting. In addition to that, we have also adopted this approach, CD45RA depleted approach, in generating multi-virus specific T cells against HPV, VZV, adeno, and BKB post-allogenic stem cell transplant. So just to give an idea of how our cells and how amending this into the clinical trial in our autologous setting clinical trial has been going on, here I'm just showing you the 12 EBV-specific T cell lines that we have generated from patients so far, and for 100% manufacturing success, including patient 1, which was a manufacturing failure to begin with, and now we have a highly antigen-specific T cells. And to see how these cells respond in patient, here I'm showing you an example of one of the lines, patient 4. So this was the antigen specificity of the line that we generated. And as you can see here, that over the different weeks after infusion of our T cells, we can see that there is an increase in the number of T cells that are secreting interferon gamma in response to EBV antigen stimulation in the patient PBMCs during their follow-up, which is really exciting. And in addition to that, we also see an increase in the frequency of T cells specific to non-EBV lymphoma antigens, so thus demonstrating epitope spreading, which has been correlated with success of T cell immunotherapy. And we observe this in our patient lines that we have infused. And however, how our cells are going to behave and how they are going to respond is really going to depend on doing further long-term follow-up analysis and historical comparisons with previous cohorts. So with this second part, I have shown you and highlighted the effects of CD45RA depletion. You can see that we can selectively expand and enrich antigen-specific T cells by our approach of CD45RA depletion, which really results in successful manufacturing of memory residing antigen-specific T cells, and also is a highly feasible clinical platform in virus-specific T cell generation. So now, next, moving on to our final point of understanding the effects of our approach and its implication in immunotherapy. So obviously, it is needless to say that we have indeed created this platform, which is highly applicable to generating virus-specific T cells. And as you have seen from our data, that it can enable us to generate highly potent T cells from patients and healthy donors, which have high potency in vitro and in vivo, and we will be able to observe it based on our clinical trial responses. In addition to all these effects, I would also like to point out this little side extra bonus that in using these allogenic third-party donor T cells, you also get this extra bonus of reduced GVSD and alloreactivity, as we know that there have been various publications, there have been various groups who have indeed done CD45RA depletion to remove highly reactive T cells in using in immunotherapy. So our approach does also aid or provides an effect in that. So with all these effects and with all these benefits, we also do know that our approach did not only restore antigen responsiveness in patient, but also overcame our manufacturing failure. So basically, just removing the CD45RA expressing subset enhanced the frequency of antigen-specific T cells, telling us that it is really not the presence of, but the lack of antigen responsiveness in our whole EBV-specific T cells. And by removing that RA-positive population, we are able to let the memory residing antigen-specific T cells to respond, which tells us that there are these various cellular subsets in the whole PBMCs when we generate our T cells for T cell immunotherapy can have different effects within each other. And we know that over the years, various groups have studied this kind of effects and have published. And just to name a few, there have been publications specific T cell subset functions. There are also been publications with a T cell interaction where Clevenov and his group had shown that there is fast-ligand interaction between naive T cells and memory T cells, which results in precocious differentiation of naive T cells in presence of memory T cells. And these are just few examples, though I did not show today, and I haven't presented how our T cells, like just by removing CD45RA positive cells, results in enriching the responsiveness of antigen-specific T cells, if it is by T cell interaction or secretion of inhibitory components. We have done we have done and evaluated some of the underlying mechanism to look into these effects, and I really hope that we're able to publish our paper soon, and you'll be able to look into this more details about the effects of RA depletion and potential mechanisms underlying the effects of RA expressing subsets. And so despite of all the success and despite all these effects that we observe with our antigen-specific T cells by CD45RA depletion in our approach, I would like to mention some of our limitations and talk about the limitations. So as I mentioned before, that we are removing stem cell-like memory, which are really known to be long-lived and cell-renewing memory cells. And I did show that our concern is diminished by long-term persistence of our EBVSTs in vitro and in vivo. Only the evaluation from follow-up and data and patient evaluation will indeed tell us if we were able to create a potent product. And also, our applicability of CD45RA depletion, where we do indeed remove TSC, is also limited by the ability of clinical grade human microbes to only CD45RA positive cells, and we cannot do so without removing TSC. There we could potentially utilize a CD95 bit selection, which would go on to select stem cell-like memory cells, central memory, effector memory, and terminal differentiated memory cells. We, however, have not utilized or evaluated CD95 bits in doing so, and it is also constrained again by the lack of clinical grade antibody to do so. Additionally, yes, our approach is highly applicable in generating virus-specific T cells and potentially tumor antigen-specific T cells, which we have not evaluated so far. But you do have to be take some concern in our strategies requiring RA positive or TSCM for persistent anti-tumor activity, meaning that if your cells of interest are residing in RA positive fraction, then our strategy might not give you the best result that you would want to for your antigen-specific T cells. And that would mostly be particularly for tumor antigen-specific T cells or new antigen-specific T cells where you're generating from third-party healthy donors, where the cells of interest might reside in RA positive fraction, and depleting CD45RA positive might hinder the success of your T cell immunotherapy. Albeit with all these limitations, I hope today I have shown you that we indeed have established a highly clinically feasible CD45RA depletion approach to enrich antigen-specific T cells for our EBV-specific T cells, which is also applicable as generating virus-specific T cells that are specific to various virus and potentially has applicability in other immunotherapy. With this, I would like to thank everyone that has helped me with this project, and specifically my mentor, Dr. Rooney, and everyone that is involved in my grad school and in our center, CAGT at Baylor College of Medicine and Texas Children's Hospital. I would also like to thank Miltony again for this opportunity to provide my work. Thank you so much.
Thank you, Dr. Sharma, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsor, Miltony Biotech, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until October 5th, 2024. Labroots will alert you via email when it's available for replay. And we encourage you to share that email with any of your colleagues who may have missed today's live event. Until next time, goodbye.